Okay, good morning, everyone. Welcome to those who are supporting Christos in the room, following, of course, all the sanitary rule. And thank you for all of you who attend from Zoom. So I'm happy today to start this defense ceremony by Christos Kotsalos. So Christos will speak 45 minutes to present his topic. I'd like just also to thank the PhD committee to be with us. So we have today online Professor Igor Pivkin from the University of Lugano, Switzerland, and Professor Gabor Zawadzki from University of Amsterdam. And we have Jonas Ladd from University of Geneva, who is the co-supervisor of the thesis with myself, Bastien Chopard. We both supervised Christos during this PhD work. So, Christos, the floor is yours, and you have 45 minutes to present your work. Thank you.
Check that everything works as expected. So great. So I'm ready to start. Bastien, thank you very much for the introduction. And I would really like to thank everybody that is attending this presentation, my PhD defense. So a very warm welcome to my PhD defense, which is entitled Digital Blood. So as you can understand from this small title, throughout all these years of my PhD, I worked on simulations of human blood. So we're trying to simulate human blood with the use of mainly supercomputers. So without further stuff to say, I would like to start my presentation. And I would like to start with some very important numbers to have in mind when we study the blood. So on average, an adult has 5 liters of blood. And in a blood drop, we can find various deformable cells like red blood cells, platelets, and white blood cells. So the red is the red blood cell, the yellow is the platelet, and the white is the white blood cell. So in this blood drop, just a few more numbers to have in mind, we can find a few million red blood cells, few 100,000 platelets, and few thousand white blood cells. So you understand what is the majority element in this small drop of blood. So these deformable blood cells, they are submerged in a liquid, which is called blood plasma. So the blood plasma is the liquid component of blood, which occupies about 55% of the whole blood volume. And the blood plasma is a Newtonian fluid, just like water. And it can be simulated just like, with Newtonian fluids. Now, from the numbers that I mentioned above, we can understand that 40 to 45% of the whole blood volume is occupied by the red blood cells. Now, this 40 to 45%, this number, we call it hematocrit. So this is the volume fraction of red blood cells. So now I would like to present you 2 clips that I remember from my childhood that are great for educational purposes. So for the ones of you that are not very aware about why we need blood, so in the left-hand side image here, we can understand a little bit better why we need blood. So blood transports oxygen, carbon dioxide, so this is the main job of the red blood cells, but also it carries various blood cells like platelets, white blood cells, nutrients to the whole body, but also bad elements like waste products, viruses, tumor cells, etc. Okay, so this is actually an image, how we can imagine blood in our blood vessels. Another very important blood cell is the platelet, which is in the right-hand side image here. And so the platelets, why they have a very important role, because they repair damaged surfaces through activation. So platelets, initially, they don't have this kind of hands that I saw in this image. So during activation, they develop these pseudopods, these blebs around them, they find a damaged surface and they go and stick towards this damaged surface. So you see here a lot of platelets that going towards the wall and repairing this damaged surface. Of course, if this process is done in excess, then later this thrombus here may detach and it can cause like a stroke or a heart attack. So in a pathophysiological case, it can create big troubles. Here I would like to show the journey of a red blood cell throughout our cardiovascular circulatory system and the importance of its deformability. So here you see a red blood cell moving in our circulatory system, and it is through the deformation of the red blood cell that we have the exchange of gases. So through the deformation, through the squeezing effect, the red blood cell releases oxygen, gets back carbon dioxide, and this is how, so everything, the pump is our heart, and then it moves around to the lungs, to the rest of the organs and the tissues in order to release oxygen and get back the carbon dioxide. So now we need to imagine that if something impairs the viscoelasticity, the deformability of the red blood cell, then most probably we will have an impaired gas exchange. So various diseases are linked with impaired red blood cell deformability. I'm going to talk a little bit more about it later. So how can we simulate blood? There are various approaches, various simulation approaches, and here the size of the problem matters a lot. So here you can see this small domain over here, which is the domain height 0.05 millimeters. And if we zoom in, we can see that the domain height, so the size of the domain, is comparable to the size of the blood cells. So here you can see the red blood cells and the platelets. So of course here we cannot neglect the particulate nature of blood, and we need to simulate to resolve all the deformable particles and all the particles of the domain. Now, if we go, for example, to this computational domain, which is this domain height is just 1 millimeter, you see that at some point the details of the domain, they don't matter a lot. So in this right-hand side image here, I have an artery, which is the diameter of the artery between 1 and 30 millimeters. Okay, and this artery has inside a stent, an artery with an aneurysm here. And here we can see that since we don't care a lot about the details, the particulate nature of blood, we can simulate it as a continuous fluid. So the message here to get is that at some point the details, the particulate nature doesn't matter anymore, and we can move from the particulate representation to the continuous representation as we have here. So the main topic of this thesis was mainly the simulation of the particulate nature of blood. So a question that naturally arises is why we need to simulate at this microscopic level. And the answer is that there are many interesting phenomena that can be better understood by simulating blood at this scale. For example, if we want to study the formation of a thrombus, of a blood clot, we need to study how the platelets and the red blood cells go towards the walls and aggregate and adhere there. Then, if we have, for example, another very interesting problem is the drug injection, where we are injecting micro or nanoparticles to the blood flow, we want to make sure that the particles go where they are intended to go, for example, towards the walls. Then there are also some other diseases like malaria and sickle cell anemia that they impair the deformability or the viscoelasticity of the red blood cells. So if we want really to understand better these diseases, we need to understand the damage of the red blood cell and see the impact on the blood flow. Also, there are some other devices like this lab-on-a-chip device for cancer cell detection. So many interesting phenomena that if we want to understand, do better designs like in this device, we need to dive deeper and resolve the particulate nature of blood. So, okay, so now let's start with the simulation of blood and how can we do it in computers. First of all, I'm going to discuss about the partial differential equations, the continuous equations that we are trying to resolve. So the blood is a complex suspension, and we need to decompose into the fluid and solid phases. So on one hand, we have the deformable bodies, like the red blood cells, platelets, and white blood cells. And on the other hand, they are submerged in the blood plasma. Okay, so to physically resolve the deformable bodies, we can do this through the elastodynamics equation. Okay, if we want to resolve the blood plasma, we need to resolve the Navier-Stokes equations. Of course, at some point, we need to couple these two phases. We need to make each phase aware of the other phase. And for this, we have the fluid-solid interaction term, which is this last term of the equations. So with this term, they can understand, the fluid can understand the existence of the deformable cells and vice versa. So now I'm going to move from this, from the partial differential equations, and I'm going to tell you how, I'm going to describe the discretized version of these equations. And for this, what we are using multiple methods to do it. We are using the finite element method for the resolution of the deformable blood cells. And of course, here we don't have only red blood cells, but we can resolve, of course, other kind of cells like platelets and white blood cells. We use the lattice-Boltzmann method in order to resolve the blood plasma. And for the coupling of the two phases, we are using the immersed boundary method. A little bit more details here. We are solving the finite elements using GPUs, graphic processing units, and we are solving the lattice-Boltzmann method and the immersed boundary in CPUs. So I would like at this point to say that we have open sourced our tool for digital bloods, and the name of this software is called Palabos NPFM. So our library is part of Palabos CFD library. And let me tell you, Palabos stands for Parallel Lattice Boltzmann Solver. So it's a solver to solve the lattice Boltzmann equations and also the immersed boundary method. And the NPFM part is the finite element solver to solve the deformable bodies. Now, Palabos NPFM is written in C++ with extensive use of MPI for parallelization of the computational tasks. And the CUDA part is mainly used for the NPFM solver that is executed on GPUs. So today, my main focus will be the NPFM solver, because actually this was my main contribution, while I was using Palabos for the lattice-Boltzmann equations and the immersed boundary, as I found them from the library from Palabos. So let me actually start now with the NPFM solver, which is a finite element solver. And here, as I said, we are trying to resolve the elastodynamics equation. So this is the continuous version, and I'm going directly to show you the discretized version. So here we are writing the implicit Euler time integration. And what we do, we write for each vertex of the discretized body, we are writing the velocity equation and the Newton's second law of motion. So the mass matrix here, M, is a diagonal matrix, because what we are doing, we are lumping the mass of the whole body into the vertices of the discretized body. So NPFM stands for nodal projected finite elements method, and this nodal part comes from the mass lumping. The projective part, I'm going to discuss about it a little bit later. So in this equation here, we have the external forces that are the forces that come from the fluid. And the internal forces is essentially the resistance of the body to the imposed external loads. And of course, these internal forces, they come essentially from the potential energy. And I'm going to talk a little bit more in details about these potential energies that we have here. So if we now take the first and the third equation and put it in the second equation, and we do a few operations, we can convert this implicit Euler time integration problem into the variational implicit Euler formulation, which is essentially an optimization problem. So what we are trying to do is to minimize this cost function. So the solution to this optimization problem leads to the trajectory and deformation of the deformable blood cell at the next time step. So let me talk now a little bit more about the potential energies. And so here we have our cost function. In order now to simulate a red blood cell or a platelet or any other deformable blood cell, we need 4 different kinds of energies: the area conservation energy, the global volume conservation the bending rigidity, and the material model. So now I observed that these three energies over here, they have a special structure. And I'm going to use this special structure in order to speed up the convergence of this optimization problem. So let me talk now about these three energies and about this special structure. So these potential energies, they are of this quadratic form. So you see that this is the quadratic form here. So Gx describes essentially the deformed state of our body. P describes the undeformed state of our body. So essentially, this potential energy penalizes how far we are from the undeformed state. So geometrically speaking, we can view this potential energy as a distance. So here is the Gx, and here is P, the undeformed state. And here M is this manifold that is formed if we take into account every possible deformed state. So a detail here that I would like to talk about is that this Gx over here is the projection. So P is the projection of Gx onto the manifold M. So again, one more time, NPFM stands for nodal projected finite elements, and the projective part of my solver comes actually from this projection. So in a sense, nature tries to find the minimum distance in order to go back to its undeformed state. Okay, so for the energies, for the potential energies that they are not of a quadratic form, here we are using this formula, which is the potential energy, and this function psi is an energy density function. What I want to say here, I don't go into the details, is that just by choosing the formula of this psi, we can simulate any material that we want. For example, we can simulate linear elasticity, Sven-Van-Kirchhoff material, Neo-Hookean materials, and the Scalac material that we are going to use for the red blood cells, as I have here. I don't want to focus into this formula, but just if we pick a different formula, we simulate different materials. So this is a continuous energy, and we do the discretization of this continuous energy using piecewise linear basis functions. And thus our finite element solver is a linear finite element solver. So now if we go back to this optimization problem, which we are trying to minimize, we can split now into the quadratic potential energies, the non-quadratic potential energies, and then let's also take the gradient of this equation. So how am I solving this optimization problem? I'm going to use a Cauchy-Newton technique to solve it. So this is an iterative procedure, and after some iterations, I'm going to find the solution to this optimization problem. And this part over here is the descent direction. So I'm moving at every step towards, I'm minimizing at every step the cost function. So this part here is a Hessian approximation. Since we are doing a Cauchy-Newton technique, this is a Hessian approximation. The better the Hessian approximation, the faster the convergence. So this H tilde is essentially the mass matrix plus this L. Okay, I will enlighten you a little bit better on this L. So L still has this formulation over here, still doesn't have a lot. So this G here comes from these quadratic potential energies. So now you have to believe me that by using this, there is a lot actually of theory of why this Hessian approximation works, but using this approximation based on the quadratic potential energies, we can have a very fast convergence to this optimization problem. So now I would like to show you a little bit of action, and here we have a stretching experiment of a red blood cell. So what we are doing here, we're taking a red blood cell and we are imposing some loads and we stretch it. And now we want to measure the axial diameter and the transverse diameter. So here you see the axial diameter and the transverse diameter. And of course, we want to validate our solver and compare it with actual experiments from the lab. So let's see here our solver, how it solves the deformation. And also here you can see that I have an actual experiment. And just to see that we are visually very close. Of course, we have the validation using the actual data from the experiments. I would like also to show you this graphical user interface that I have developed, which is, so this means that I can use the NPFM solver as a standalone version. And for this, I used Rhino and Grasshopper. Rhino is this window over here. It's a computer-aided design software. And Grasshopper, which is this window over here, it's essentially a plugin to Rhino. So what we can do here, we can, through this graphical user interface, we can load any kind of mesh. We can set up the material. We can set up the forces, and then we can run the NPFM solver. And so here you can see a deformed red blood cell. So actually, it's a very nice graphical user interface where people can pick any kind of material and check if this is a valid material. Also, they can do some changes, modifications in the NPFM solver and visually inspect if they have bugs or not. Also, this is an open source, and you can find it in Palabo's CFD library. Another deformation that I really like, in this case, actually, we have also the fluid-solid interaction. It's when a red blood cell flows inside the capillary. So this capillary, this small vessel, is about 10 micrometers, and the diameter of a red blood cell is about 8 micrometers. So you see that when it flows through this capillary, we have this parachute-like shape. And also here I have the actual experiment that I performed. And of course, here also we can visually inspect this shape with an actual shape from an actual experiment, from an in vitro experiment. But okay, so far so good, but I just showed you single red blood cell experiments. Now the idea is to put a lot of red blood cells and platelets in one computational domain and do some interesting simulations. So here we have a lot of solvers to combine. So we have the fluid solver executed in CPUs, the fluid-solid interaction, again executed on CPUs, and we have the solid solver executed on the GPUs. So now the question is how we are going to do the load balancing, dispatch the load into the available CPUs and GPUs. So let me start with the fluid solver part, the lattice-Boltzmann solver of our framework. And so here the fluid is resolved on a static homogeneous grid. So what we are doing here, we're taking the computational domain, subdivide it into non-overlapping boxes, and we assign each box to the available CPU cores. Also, every CPU core is linked to an MPI task. MPI is for the parallelization. It's a library for parallelization. Now, regarding the NPFM part, the solver, the FEM solver for the deformable blood cells. So the MPI tasks, like the MPI task J and K, that are linked to a GPU, they can now deal with deformable blood cells. And so here, what we are doing, we are distributing once at the beginning, so in a static matter, the available blood cells to the available GPUs. So in our case, we have two GPUs, one linked with MPI task J and the other linked to MPI task K. So these bodies, they can be spatially everywhere. For example, let's take the red blood cell with ID 1. This spatially is located in a memory space that is handled by the MPI task K, as I have here. So now the actual NPFM solver is located in MPI task J, so there needs to be, for the coupling of the two solvers, a kind of communication. As I'm showing here, we need to pass some data from task K to task J, and then from the CPU to GPU. And this we are doing it using the MPI point-to-point communication, where, for example, we have to transfer from the fluid solver forces and collision data, so from MPI task K to MPI task J, and then we need to overload from the CPU to the GPU, do the computations, and when we are done with the solution of the deformations and trajectories, we need to communicate it back through the same stream. A little bit further on the GPU acceleration. So here we have, again, the NPFM solver, which is linked to a GPU, and it has to deal with M blood cells. So this is a GPU here, which is an array of streaming multiprocessors. So these boxes here are the streaming multiprocessors. And these boxes, essentially, they group together the GPU cores. So now the idea is to assign one CUDA block per blood cell, and then dispatch these CUDA blocks to the available streaming multiprocessors. Okay, and why we fit the bodies inside the streaming multiprocessors? Because we want to take advantage of a very fast shared memory that can be found in here. And we want also to take advantage of the thread synchronization provided inside the streaming multiprocessors. So now if we put all these pieces together, throughout these years, we ran a lot of experiments. For example, we ran we ran different Couette and Poiseuille flows. So here you can see 3 different Couette flows of different domain sizes. So I don't want you to focus into the detail, but just to see the simulations that we got. I will zoom in the next slide. And also let's see a little bit closer a Poiseuille flow. So this is a flow inside the tube. And this is, actually this is a simulation with only red blood cells, but here also you see that the yellow parts, the yellow bodies are the platelets. So we also focused in pathological conditions, meaning that the red blood cells were more swollen than the healthy red blood cells. And here you can see again a Couette flow where we have a moving upper boundary. And this is actually the simulation, the flow that we are expecting to see. And also in this right-hand side image, we are orbiting around the domain. Again, it's the same simulation, but we are orbiting around the domain just to see what we get. Of course, if we put all these pieces together, at some point we have to wonder if our framework, the Palabos NPFM, is performing well. So for this, we need to run some performance measures, tests. And for this, we used the Pitch Giant supercomputer located at the Swiss National Supercomputing Center. And it's one of the fastest supercomputers in the world. So this supercomputer has 5,700 GPU nodes. And every GPU node, it has 12 CPU cores and 1 NVIDIA GPU. So now, in order to test the performance of our computational framework, we did this through a weak scaling study. So in the weak scaling study, we are starting from this small domain over here, and we are gradually increasing the size of the computational domain. But as we increase the size of the computational domain, we proportionally increase the amount of hardware that we are using. So we are resolving this small domain using 5 GPU nodes. And in an 80 times bigger domain, as it is this, we are using 80 times more GPUs. So in this case, we are solving this domain using 400 GPU nodes. So now, since we are proportionally increasing the amount of hardware that we are using, if I measure the time to solve the small problem and the subsequent times to solve the bigger problems, essentially these times, they should be the same. And this is how we quantify the efficiency of our framework. So the efficiency is Tn0 over Tn. And this is actually now what I'm going to present you here. And we see that the efficiency of our computational framework starts apparently from 100% and has a small drop. And it is between 75 to 80% efficiency. So here you can see that this efficiency is for the biggest domain, 80%. And we resolved also 2 different hematocrits, 35% and 45%. And you can see that the efficiency drop is not actually that much. So about 80% efficiency is a great efficiency. And so after this test, we were quite happy about the performance of Palabos NPFM framework. So the last part on the computational aspect of my solver is the cell packing. So before start doing these simulations, the first thing that we need to do is to pack the cells to create a cell field. So we need to put the blood cells inside our domain, initialize the field, and then run the actual simulation. So here, what we are doing, we are going actually to take advantage of the implicit nature of the MPFM solver. So the implicit nature means that it is an unconditionally stable solver, and through this unconditional stability, we can resolve extreme deformations and interpenetrations. So what I'm doing here, I was randomly initializing the positions of the blood cells. So you can see in this image that there are severe interpenetrations. And actually, if I run now the MPFM solver for a few thousand iterations, you will see how efficiently the MPFM solver solves these extreme interpenetrations and deformations. So now this is the image. This is the initial cell field that we are going to start the actual simulations. So now enough with all the computational aspects of my Palabos MPFM solver, and let's see some action. And at some point we have to wonder if we are resolving actual physics or we are doing computer graphics. And for this, we have the in silico part, the simulations, and we are going to use them along with in vitro actual experiments from laboratory and see if our solver gives meaningful results. So one of the first projects that we did was a collaboration with the Laboratory of Experimental Medicine led by Professor Karim Zouaoui Boujeldia. And the title of this project was Spherization of Red Blood Cells and Platelet Margination in COPD patients. COPD stands for Chronic Obstructive Pulmonary Disease, and it is mainly linked to smoking. So smokers most probably at some point may develop COPD. So the first part of this study, what we found out is that in COPD patients, the red blood cells, they are more spherical than healthy people. So you see here, I have some simulations with healthy red blood cells, which is like a donut-like shape. But as we have COPD patients here, the red blood cells are more spherical, more swollen. So now we want actually to see if the impact of this shape change in platelet margination. And what is platelet margination? It is the migration of the platelets towards the vessel walls. And this is because of the existence of the red blood cells. So because of the red blood cells, the platelets are pushed towards the walls of the vessel. And this phenomenon is called margination. Now we want to quantify the shape change to quantify the impact of shape change on platelet margination. And what we found out, both using the in vitro part and the in silico simulations, the in silico part, we found out that the platelet margination is more pronounced in patients with COPD. So more spherical red blood cell shape pushes more the platelets towards the vessel walls. So this means that if we have higher margination, this leads to higher concentrations of platelets towards the walls. So more cardiovascular problems in the sense that we may have an excessive thrombus formation. And again, from one of the first slides, if a thrombus detaches, it may cause a heart attack or a stroke. So this was one of the important first projects where we are using both the in silico and the in vitro part in order to validate our solver, but also do more in-depth investigations of problems. Now, the last project actually that we dealt, it was built upon a study that Bastian and Karim did back in 2017. So back in 2017, they published a paper called The Physical Description of the Adhesion and Aggregation of Platelets. So the in vitro part of this experiment was using the IMPACT-R platelet function analyzer medical device. So IMPACT-R is this medical device, is this simple cylindrical device, where we put here blood. So we put here red blood cells and platelets with blood plasma. There is here this rotating upper disc. And what we want to study is essentially how the platelets deposit towards this bottom wall. So So essentially, we have again margination, migration of platelets towards the walls of the system. And we want to study essentially this kind of images, how platelets aggregate and adhere to this bottom wall. Now, if we focus in a small domain of the IMPACT-R device, the in silico counterpart is something like this. So in this domain here, we have essentially the red blood cells, the activated and non-activated platelets, and albumin. Albumin is a protein that competes in adhesion and aggregation. So in this bottom layer, in this bottom wall, we will have activated and non-activated platelets adhering and aggregating, but also albumin. So here we have the in silico experiments. We have also here the in vitro experiments, and we want to do a comparison. So back in 2017, Bastian solved this problem. So he replaced the complex dynamics of these particles with a diffusion, with a diffusive process. So the margination traditionally is explained by red blood cell enhanced C-reduced diffusion. So this process is a diffusion process. So since this is a diffusion process, we can use this equation, which is the diffusion equation, in order to solve the dynamics in the bulk of the IMPACT-R device, and then study the deposition in this plate. So this process here, this diffusion process, is equivalent to random walks with velocities following a Gaussian probability density function. So let me talk. Random walks is the random walk is essentially a random movement of a particle as a result of continuous collisions with the surrounding particles. So if here we had this platelet performing a random walk, we could see something like this. So it's moving around after colliding with the surrounding particles, and at some point it's going to go back to this. So what Bastian found was that the only way to match the experimental results was to use here a diffusion coefficient of the order 10 to the minus 8. But this diffusion coefficient was 1,000 times more than the Zidny-Colton formula, which is a formula that gives the diffusion coefficient as a function of the hematocrit and the C rate. Okay, but also this diffusion coefficient was 1,000 times more than any other source in the literature. And also we tried through Palabos NPFM to quantify the diffusion coefficient, and we were finding something completely different than this value. So now we wanted to understand why this is happening. So now this is the last part of my talk. And at some point we questioned and we said, okay, either the Zidny-Colton formula is wrong. Actually, the Zidny-Colton formula is based on experimental and theoretical studies, so there is not a lot of ground on saying that this formula is wrong, because it was fitted on actual experiments. And then another option is that the platelet transport physics, they do not follow a diffusion process. So this equation, that it is traditionally considered to be the way that platelets migrate, is not accurate. So now our hypothesis is that platelets, they do not diffuse in a normal way. So they do not follow a Brownian-like a Gaussian random walk. So the probability of their velocity, the probability density function of their velocity is not proportional to this Gaussian kernel, but they obey a Levy flight. So their probability, the probability density function of their velocity follows this power law, this fat tail distribution. So let me show you in this slide why this is very important. So here I'm going to show you two random walkers, two platelets. One platelet, the red platelet here, will follow a Levy flight, and the blue platelet is going to follow a Gaussian walk. So let's observe here how they explore the space. So the red is the Levy flight, and the blue is the Gaussian walk. So you see these huge jumps of the red particle, and essentially the red particle explores the domain in a completely different way than the blue particle. Okay, and I would like here to mention as well, to highlight that these two walkers, they have the same average jump length. Okay. Also, I need to highlight that this exponent here, alpha, it should be less or equal to 2 in order to observe this kind of extreme events, velocities. And if it is above 2, essentially we are closer to a Gaussian random walk. So actually, to try to distinguish between a Levy flight and a Gaussian walk is extremely important for the platelet transport physics. So now, let me also talk a little bit more about these extreme events and where is the problem. So in our normal simulations, let's track here individual platelets, and we can extract the mean square displacement. So the mean square displacement, which is this formula over here, encodes how fast the platelets explore the space. So essentially, it encodes how fast they move away from their initial position. So essentially, the slope of the mean square displacement is traditionally linked to the diffusion coefficient of the platelets. So if the platelets follow a diffusion process, so if they follow this kind of equation, we can use immediately this value from our simulations. This is actually what we did at the beginning. But then we can rewrite the mean square displacement as a function of the variance. So this is the variance of the velocity of the platelets. But if the platelets, they follow a Levy flight with a probability density function following this power law, and alpha is less or equal to 2, the variance is infinite. So with infinite variance, we have essentially an undefined mean square displacement. And of course, if the platelets follow a Levy flight, we do not have a diffusion process anymore. And of course, these kind of equations, they cannot be applied anymore. So now we tried to see if we can find these Levy flights in our simulations. On one hand, we have the IMPACT-R platelet function analyzer. On the other hand, we have this simplified model that Bastian wrote back in 2017. But on the other hand, we have also our Palabos NPFM simulations. So now the idea is that let's run a lot of this kind of simulations, essentially to simulate this box. Let's actually track individual platelets, extract the mean square displacement, the diffusion coefficient, and also we want to check the underlying probability distribution density function of the platelet velocity. And we want to see if indeed they follow a Gaussian walk or a Levy flight. Okay, so the first step that we followed, we ran different experiments, as you can see here. Let me bring the cursor. Okay, we ran different experiments in larger domains as well. And we extracted here in the y-axis the diffusion coefficient. So the diffusion coefficient as a function of the domain, the wall-bounded size. Okay, and what we observed is that the diffusion coefficient, or essentially the mean square displacement, diverges with increasing sample size. So as we go to sample sizes, to larger sample sizes, we had an increasing mean square displacement instead of a converging behavior, because as we increase the sample size, we expect all the moments to converge somewhere. But this is not the case. The striking part is that the diffusion coefficient that we extracted, so here we take the mean square displacement, we extract the diffusion coefficient. This diffusion coefficient is compatible with the Zidnik-Orton formula. So let me say that the mean square displacement will always be finite, because in our systems here, we have a finite number of platelets. So what we can do, we can take these systems, we can extract the diffusion coefficient, and since it is compatible with the Zidnik-Orton formula, we could say that everything is okay. But if we dive a little bit deeper and see how the mean square displacement behaves with increasing computational domain, we will see a divergent behavior. So a behavior that is more compatible with Levy flights and not with diffusion processes. So this is, and then what we did, we went in every simulation that we ran. So here we have our computational domain with the platelets and the red blood cells. What we did, we gathered all the platelet velocities per simulation. We plotted the histogram of the velocities, and what we tried do is to fit this kind of power law on the tail of the distribution of our histogram. So what we found out, initially our hypothesis is that they follow a Levy flight with an exponent less or equal to 2. So what we did, we ran more than 60 Palabos and BFEM simulations. We had domains of different sizes. We used various types of platelets, like activated and inactivated platelets, 2 different hematocrits, 35% and 20%. And what actually we found out is that the particles, they obey a Levy flight with an alpha, which is about 1.5 plus minus, depending the case that we are dealing with. So indeed, our hypothesis seemed to be correct. Then what we did, we went back to this simplified model that Bastian developed back in 2017. And we said, okay, we have here these processes. And instead of a diffusion process, we can use here a random walk. But the random walk now will follow a Levy flight, this power law distribution with alpha 1.5 plus minus 0.3. So now here we have a platelet following this random walk with these extreme events. So we don't have a diffusion process. Essentially, we are reinterpreting the platelet transport physics. And by simply using this random walk instead of the diffusion process, we could now match the experiments that we had from the actual IMPACT-R device. So without tuning, for example, the diffusion coefficients, the diffusion coefficient would be 1,000 times more than what we were finding. And now this is actually my last slide. A possible explanation of this kind of extreme events, of these extreme velocities. This is a slice from my computational domain at time 0. And this is also a slice at another time, few time steps afterwards. And you can see this heterogeneous RBC distribution. So here we have these big voids. So what actually some researchers said before us is that these voids here, they can be used as express lanes for platelets. So platelets, they can move faster into these voids, and they can produce this kind of extreme events, velocities, which essentially result to this Levy flight physics. And actually, this is my last slide. And I would like to thank you very much for your attention.